Okay, welcome back. We're going to continue reviewing Chapter 3. Trying to remember where we left off here. Definitely did all this. We did lipids. Okay, proteins. Here we go with proteins. Proteins account for more than 50% of the dry mass of most cells. They have a lot of different functions, very diverse functions, including defense, storage, transport, cellular communication, movement, and structural support. Enzymes are proteins that speed up chemical reactions. Here you have an enzyme binding to two reactants and releasing products. So this would be a reaction where a larger substance gets broken down into two smaller ones. You have defensive proteins. We just finished the immune system. So defensive proteins are proteins that protect us against disease and infection. Example would be your antibodies. You have storage proteins. Function would be the storage of amino acids. Examples include casein, the protein of milk, which is the major source of amino acids for baby mammals. We've learned a lot about transport proteins, those proteins that are embedded within the cell membrane that allow the transport of molecules into or out of the cell. One example would be hemoglobin, the iron-containing protein of vertebrate blood, which transports oxygen from the lungs to other parts of the body. Another example of your transport protein would be, for example, your sodium-potassium pump. Or the proteins that are embedded within the mitochondrial membrane or the thylakoid membrane that allow the transport of hydrogen ions. You have hormonal proteins. Function is to coordinate an organism's activities. For example, insulin is a hormone secreted by the pancreas, causes other tissues to take up glucose, thus regulating blood sugar concentration. The opposite of that would be glucagon. Those are antagonistic hormones. Antagonistic meaning they have opposite functions. You have receptor proteins. Here this is showing a cell of the nervous system, your neuron, which is releasing signaling molecules. You know those signaling molecules as neurotransmitters. And those neurotransmitters will bind to a receptor protein on the postsynaptic cell. Remember that those signaling molecules are very specific to the receptors. You have contractile and motor proteins such as actin and myosin, which make up your basically your muscle tissue. And then you have structural proteins which provide support, such as keratin, which is the protein of your hair, horns, feathers, and other skin appendages. Life would not be possible without enzymes. Enzymatic proteins act as catalysts to speed up chemical reactions without being consumed by the reaction. Polypeptides are unbranched polymers built from the same set of 20 amino acids. A protein is a biologically functional molecule that consists of one or more polypeptides. Amino acids are organic molecules with carboxyl and amino groups. Amino acids differ in their properties due to differing side chains called R groups. Okay, so here is the basic structure of an amino acid. You have your amino group over here. You have your carboxyl group over here. Your central carbon is called your alpha carbon. There's a hydrogen off of your central carbon. And then also off of that central carbon is what's designated as R, which stands for your side chain. So here are some of your amino acids. These are amino acids that are hydrophobic because they have non-polar side chains. So notice that non-polar side chains consist primarily of carbons and hydrogens, which are non-polar covalent bonds. They create non-polar covalent bonds, thus making the side chains hydrophobic and water-fearing. You have amino acids that are hydrophilic because they have polar side chains. One of the elements that you see appearing a lot here is oxygen. Oxygen is a highly electronegative element. which causes bonds that it forms to be polar covalent, which leads to that partial charge on both elements, a partial negative on the oxygen, and a partial positive on the hydrogen. That makes those side chains polar and therefore hydrophilic. And then we have acidic side chains, or acidic amino acids, which are negatively charged, and basic amino acids, which are positively charged. So you can see that the side chains carry charges in all of these examples. Amino acids are linked by peptide bonds. A polypeptide is a polymer of amino acids. Polypeptides range in length from a few to more than 1,000 monomers. Each polypeptide has a unique linear sequence of amino acids with a carboxyl end, which is the C-terminus, because the carboxyl has a carbon, and an amino end called the N-terminus, because the amino end has a nitrogen. This is the formation of, in this case, looks like just a tripeptide. So here we have a peptide bond, which occurs between the carbon of the C-terminus end of one amino acid and the nitrogen from the N-terminus of another amino acid. And then through condensation synthesis or dehydration synthesis, what happens here is a hydroxyl group gets removed from this amino acid, from the carboxyl end, and a hydrogen gets removed from the amino group of this amino acid, forming water and creating another peptide bond between the carbon and the nitrogen. A functional protein consists of one or more polypeptides precisely twisted, folded, and coiled into a unique shape. So here's what that might look like. The sequence of amino acids determined genetically leads to a protein's 3D structure. A protein structure determines its function. That's one of the underlying themes of biology. So here we have an antibody protein that's reacting with a protein from the flu virus, so there's great specificity between the two. Proteins are very diverse, but share three superimposed levels of structure called primary, secondary, and tertiary structure. A fourth level, or quaternary structure, arises when protein consists of more than one polypeptide chain. The primary structure of a protein is just simply the sequence of amino acids, which one comes first, which one comes second, third, fourth, and so on. That's determined genetically. Secondary structure found in most proteins consists of coils and folds in the polypeptide chains. So as you recall, those are your alpha helices and your beta pleated sheets. And then your tertiary structure is determined by interactions among various side chains. Those include hydrophobic interactions, ionic bonds, disulfide bridges, hydrogen bonds. And then lastly, quaternary structure results if you have more than one polypeptide chain interacting together with those same bonds as the tertiary structure. So here's the primary structure. It's just the sequence of amino acids from the beginning to the end. Here's your secondary structure, which results from hydrogen bonds of your backbones of your amino acids, which leads to either alpha helices or beta pleated sheets, both of which can be found in the same protein or polypeptide. Here's your secondary structure again. And then your tertiary structure and your quaternary structure. Your tertiary and your quaternary structure, these are the bonds that hold those together. Notice that these bonds occur between the side chains of your amino acids. So here we have a hydrogen bond. Here we have hydrophobic interactions and van der Waals interactions because all of these are non-polar bonds. Here we have an ionic bond occurring between a negatively charged oxygen and a positively charged hydrogen, different from a hydrogen bond because this results from partial charges on the elements. These are full charges. And then, of course, your disulfide bridge, where you have an interaction between two sulfurs. And then here is hemoglobin, which exhibits a quaternary structure. It's hard to tell, but there are actually two separate polypeptides here that are interacting, an alpha subunit and a beta subunit. Primary structure is the sequence of amino acids on the polypeptide chain. A slight change in primary structure can affect a protein's structure and its ability to function. Sickle cell disease is an inherited blood disorder, and it results from a single amino acid substitution in the protein hemoglobin. So if you look here, here's the normal sequence of amino acids, the normal primary structure. Here is sickle cell. Notice that at your sixth position, you have an incorrect amino acid. You have valine here. That is the incorrect amino acid. This leads to a shape change, and it leads to the blood cells being sickle-shaped. In addition to primary structure, physical and chemical conditions can affect structure. Alterations in pH, salt concentration, temperature, or other environmental factors can cause a protein to unravel. This loss of a protein's native structure is called denaturation. A denatured protein is biologically inactive. Okay, so here's the normal protein structure. Now remember, with your proteins, for example your enzymes, because of the tertiary and or the quaternary structures, that leads to very specific shapes in the molecule. For example, that leads to a groove in the enzyme called the active site. And of course the active site is where the substrate binds to the enzyme. Well, if that protein, that enzyme unravels, then that active site will no longer be there. There will no longer be that specifically shaped groove in the protein to which the substrate can bind. Now, remember that the structure, all the levels of protein structure, are genetically determined. So if we reinstate the original environmental conditions, that protein will renature. It'll go back to its native state because the native state of the protein is genetically determined. We skipped over this. Okay, nucleic acids. The amino acid sequence of a polypeptide is programmed by a unit of inheritance called a gene. Genes are made of DNA, a nucleic acid made of monomers called nucleotides. There are two types of nucleotides, deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. DNA provides directions for its own replication. DNA directs synthesis of messenger RNA, and through mRNA controls protein synthesis. So remember that your DNA is found within the nucleus. Your mRNA is basically a complementary copy of the DNA. It will take the message that's found within the DNA into the cytoplasm and to the ribosome, where protein synthesis will occur. The ribosome will read the mRNA, and other molecules will bring in the correct amino acid corresponding to the particular mRNA codon, and a polypeptide or a protein will get built from the ribosome and the mRNA interacting together, of course along with your transfer RNA, which brings in the amino acids. Nucleic acids are polymers called polynucleotides. Each polynucleotide is made of monomers called nucleotides. Each nucleotide consists of a nitrogenous base, a pentose sugar, and one or more phosphate groups. Okay, so here is one side of a DNA molecule. So this is a sugar. The little circles are, not sugars. The little circles are phosphates, and the green molecules are the sugars. So it's the backbone, or the rung of the ladder of the DNA, is a phosphate sugar alternating backbone. And then in the middle, you have the nitrogenous bases, which consist of your pyrimidines, cytosine, thymine, and uracil, and your purines, which notice are double sugar rings, adenine and guanine. If we are talking DNA, DNA has deoxyribose. RNA has ribose. Notice the only difference is right here, okay, on your second carbon. In deoxyribose, it's just a hydrogen, so deoxy without oxygen, so it's missing an oxygen right there, whereas in ribose, you have an OH group there. Each nitrogenous base has 1 or 2 rings that include nitrogen atoms. The nitrogenous bases in nucleic acids are called cytosine, thymine, uracil, adenine, and guanine. Thymine is found only in DNA. Uracil is found only in RNA. The rest are found in both DNA and RNA. The sugar in DNA is deoxyribose, in RNA it's ribose. A prime is used to identify the carbon atoms in the ribose, such as the 2nd prime carbon or the 5th prime carbon. So remember that when we count the carbons, we always start to the right of the oxygen and we go clockwise. So this is your 1st carbon, 2nd, 3rd, 4th, and your 5th one comes off of the top. Adjacent nucleotides are joined by covalent bonds that form the OH group on the 3' carbon of one nucleotide and the 5' phosphate on the 5' carbon of the next. These links create a backbone of sugar phosphate units with nitrogenous bases as appendages, as you saw in the picture. The sequence of bases along a DNA or mRNA polymer is unique for each gene. RNA molecules usually exist as single polypeptide chains. DNA molecules have 2 polynucleotides spiraling around an imaginary axis forming a double helix. In the DNA double helix, the 2 backbones run in opposite 5' to 3' directions from each other. This is referred to as the anti-parallel nature of the DNA double helix. One DNA molecule includes many genes. The nitrogenous bases in DNA pair up and form hydrogen bonds. Adenine always bonds with thymine, and guanine always bonds with cytosine. So A with T and C with G. This is complementary base pairing. Complementary pairing can also occur between 2 RNA molecules or between parts of the same molecule. In RNA, however, there is no thymine. Uracil is the replacement for thymine, so uracil matches up with adenine. So here is the structure of your double helix of DNA. Notice that it is anti-parallel. On this side, you have the 5'. On this side, you have the 3'. Down here at the bottom, it's the opposite. And then this is just showing that in transfer RNA, the nitrogenous bases can pair up with each other within the same molecule. The linear sequences of nucleotides in DNA molecules are passed from parents to offspring. 2 closely related species are more similar in DNA than are more distantly related species, and molecular biology can be used to assess evolutionary kinship.